the general characters of the rhodophyte. जैसा कि मैंने आप लोगों को कल भी बताया था कि जो एल्गल क्लासिफिकेशन है, it is based on the crude classification or the primitive classification. It is based on the thallus color, the type of flagella, life cycle, cell wall characteristic features, then cell type, flagella, stigma. There are many other characters. Reserve food, nature of the reserve food material, then type of the pigments which are found in algae. So these characters are responsible for classification of the algae. But as we know that the red algae is due to the presence of red pigment which are found in excessive amount in such a way that they hide the green color of the chloroplast. So in red algae, there are two pigments, R-phycocyanin and R-phycoerythrin. It is specifically R-phycoerythrin pigment which hide the green color of the chlorophyll, which makes the thallus of red algae red in color. Red in color. The chlorophyll B is replaced by the chlorophyll D. I told you it is a doubtful statement.
Yes, sir.
Right. The chief xanthophyll pigment means the carotenoids. The chief carotenoid or the xanthophyll is teraxanthin, or you can say taraxanthin. Okay.
Okay, sir.
So this is about why the red algae are red in color. The red algae are characterized by the absence of the flagellated motile stage. In any stage of the life cycle of red algae, whether it is vegetative stage or reproductive stage, either gametophyte or sporophyte or tetrasporophyte or carposporophyte. In any stage of the life cycle, you cannot find any flagellated or motile structure in its life cycle. That is very important feature of the red algae. And in this feature, it is also similar to the blue-green algae or the cyanobacteria, because this statement will be repeated in there also, because in cyanobacteria also, you will not find any flagellated stage in their life cycle at all. So in this respect, red algae shows similarity with the blue-green algae, that there is no motile stage or no flagellated stage.
Flagellated.
The main food product of the red algae is the Floridian starch. कल मैंने आप लोगों को बताया था starch, तीन तरह के starch है ना, simple starch, mixophycin starch and Floridian starch. So the main food product is Floridian starch, or it is also known as floridoside and manoglycerates. These are the reserve food material. In addition to the cellulose and pectin, the chief characteristic component of the cell wall is polysulfate esters. Besides cell wall, in cell wall, besides cellulose and pectin, they contain polysulfated esters, and the cell wall consists of rigid components such as the microfibrils and mucilaginous matrix. They contain special polysaccharides, which is known as agar and carrageenans.
Yes, sir.
Agar and carrageenans. These are sulfated polysaccharides, which is found in cell wall of the red algae.
Cell wall of red algae.
They have very important. They have very important economic importance.
Yes, sir.
The thallus size of the red alga, it ranges from the unicellular up to 1 meter or 2 meter long. Some genera are, particularly the marine one, they are up to 1 meter or 2 meter long. For example, Gaigartina and Saigimania. These two genera may reach up to the length of 1 meter or 2 meter tall, while some members are unicellular also. So they are they range from microscopic unicellular up to the 2 meters in length. Except a few unicellular forms like Porphyridium. Porphyridium is a unicellular red alga. The great majority of the red algae are filamentous in thallus organization. So majority of the red algae, they show filamentous type of thallus organization. For example, Goniotrichum, Gracilaria, Kappa ficus. These are the parenchymatous forms. Means these are
Yes,
very
sir.
large.
Pseudo.
They have large thallus. Gracilaria, Kappa ficus. They show pseudoparenchymatous type of thallus organization, whereas Dumonsia and Hemicladia and Porphyra.
Parenchymatous.
Yeah, these are the parenchymatous forms.
Yes, sir.
Most of the multicellular red algae, the vegetative cells are connected by the pit connection. Here you can see in this diagram, there is a pit connection. This pit connection you can also find in fungi also. Can you tell me the name of the group of fungi where we get the pit connection?
So pit connection.
फंजाई के भी एक ग्रुप में पिट कनेक्शंस पाए जाते हैं, that is known as Dolipore septum, and it is found in members of the Basidiomycotina.
Okay, sir.
Rust and smuts. So in red algae, the filamentous red algae, they contain the pit connections which connect two adjacent cells. Pit connections are two types: primary pit connection and secondary pit connection. Primary pit connection, they are present in one single filament. But if the thallus is multi-axial, in that case, two adjacent filaments are connected by the secondary pit connection. And this pit connection serve as a passage for the cytoplasmic material flow between the adjacent cells.
Adjacent cells.
This is found only in red algae, not in any other group of the algae.
Okay, sir.
This is the important characteristic feature of the red algae. The sexual reproduction is highly specialized and advanced oogamous type.
Oogamous type.
Male sex organs in red algae, they are spermatangia. In each spermatangium develops a single non-flagellated, non-motile male gamete known as spermacium. So spermacium is the name of male gamete, and the gamete is formed in a structure known as spermatangium. So inside the spermatangium, there is formation of one spermacium, right?
Yes, sir.
So the female sex organs, they are known as carpogonia. So the term carpogonia and spermacia are used only for red algae, not for any other group of the algae. So the female sex organs, they are known as carpogonia. Each carpogonium is a flask-shaped structure having a long neck-like structure known as trichogyne, whereas the base is swollen. I can show you the structure. So here you can see the carpogonium. Carpogonium has long neck-like structure. You can see this neck-like structure is known as trichogyne, and lower part is swollen, flask-like. So this whole structure is carpogonium, which is the female reproductive structure. Right here, you can see the structure of the spermatangia. So spermatangia, these are the apical cell of this branch, this lateral branch of. There are certain specific branches which are found in male gametophytic plants of the red algae, and these branches are known as trichoblast branches. On these trichoblast branches, we can find the spermatangia. The apical cell of the trichoblast branches, they form the spermatangia, and each spermatangium produce only one male gamete known as spermacium. These spermacia means the male gamete, they are without any flagella. No flagella is found. That is very exception in whole plant kingdom where the gametes are without any flagella or cilia. Otherwise, in all plants, the flagella, the reproductive structures, particularly the male reproductive structures, are either flagellated or ciliated, which help in locomotion to reach up to the site of female reproductive structure or the egg cell. But here it is the exception in plant kingdom where the male gametes are without any locomotory organ.
So, sir, how does the fertilization occur?
I will tell you. During fertilization, the non-motile male gamete are taken up to the trichogyne with the help of water current,
Okay.
because they are aquatic in nature. And when
Yes.
the spermatia are released from the spermatangium, they reach up to the carpogonium
Carpogonium
passively
with the help of
through
water.
the water current.
Okay, sir.
During fertilization, post-fertilization
Post-fertilization.
changes or stages are also highly elaborate and characteristics of the red algae. Though they exist in many variations, many gonimoblast filament develop from the fertilized carpogonium in many cases. So the female reproductive structure, which is known as carpogonium, it contains the egg cell. After fertilization, this carpogonium produces many filaments coming out from the carpogonium, and these filaments are known as gonimoblast filament, right? So the gonimoblast
Yes, sir.
filaments develop from the carpogonium, which is the female reproductive structure of the gametophyte, right?
Yes, sir.
So carpogonium produces gonimoblast filaments after
Filament.
fertilization, right?
Yes,
The
sir.
tip of this gonimoblast filament start function as a carposporangium. जो tip होता है in
Yes,
gonimoblast
sir.
filament का,
Filament भी.
it function as a sporangium, which is known as carposporangium, and each carposporangium forms a single. Each carposporangium forms a single carpospore, and this carpospore further germinate either into a gametophytic phase or it may develop into the tetrasporophytic phase. So this is the fertilization. This is the female reproductive structure, carpogonium. From this carpogonium, after the fertilization, these filaments are forming. You can see numerous filaments are developing from this carpogonium after the fertilization. This is the carpogonium, and many filaments are coming out from this carpogonium after the fertilization. The tip of the carpogonium develops a sporangium known as carposporangium, and in each carposporangium, a single spore is found, which is known as carpospore. So obviously, this carposporangium, and gonimoblast filament, they all together constitute the carposporophyte. इसी को हम लोग कार्पोस्पोरोफाइट कहते हैं, जो कि किससे बनता है? इसी कार्पोगोनियम में इसके एग सेल का फर्टिलाइजेशन के बाद बनता है. फर्टिलाइजेशन के बाद फिलामेंट्स. इसी कार्पोगोनियम से क्या बना? कार्पोस्पोरोफाइट. कार्पोस्पोरोफाइट किसका बना होता है? गोनिमोब्लास्ट फिलामेंट, कार्पोस्पोरेंजियम एंड कार्पोस्पोर. क्योंकि ये पूरा जो कार्पोगोनियम है फीमेल गैमेटोफाइट पे है, तो ये जो कार्पोस्पोरोफाइट बना, ये कार्पोस्पोरोफाइट भी किसके ऊपर डिपेंडेंट है अपने न्यूट्रिशन के लिए? फीमेल गैमेटोफाइट पर. In higher forms, for example, Polysyphonia, they produce tetraspores and tetrasporophyte. These carpospores develop into tetrasporophyte, and tetrasporophyte forms the tetraspore, but it is found in higher red algal form. Most of the members show diphagic or triphagic life cycle. So this is about the general characters of the red algae. Let us go something important features, other important features in bit detail. Yes, sir. The habitat where we find the red algae. The red algae, they are basically marine in habitat, but some forms of the algae, red algae, they are known from the freshwater habitat also. The freshwater algae, red algae, are Lemania, Batrachospermum, Compsogone. These three are the freshwater red algae which are found in flowing water or in streams or rivers. Right. Yes, in the Batrachospermum is also found in ponds also. A few species of Gracilaria. Gracilaria is basically a marine, but it can also found growing in brackish water, that is the meeting point where the fresh water meets with the marine water, particularly the estuaries, जहाँ पर नदियाँ rivers में मिलती हैं. In
Yes,
estuaries,
sir.
we find the brackish water where the gracilaria is known to grow in such a habitat. Most of the members, they are marine, and they found in intertidal or sublittoral zone of the sea coast. Some red algal species, they are found in terrestrial habitat also, like they are found growing attached on soil, rock. There is unicellular red alga, porphyridium, which is found in moist soil.
Soil.
Unicellular red alga, porphyridium. Many red algal members, they show high degree of the epiphytism and parasitism. Means they are found growing attached on other plants, and some members of the red algae, they are parasite on the other group of the plants. Right?
Yes,
For
sir.
example, in case of parasitism, Gelidiocolax is host plant Gelidium. Gelidium is the host plant, and on this Gelidium, the parasitic red alga is
No,
Gelidiocolax. Other parasitic forms are Polysiphonia species. This Polysiphonia, which is found growing attached on another red alga, Escophyllum nodulosum, right? Ceramium species is found growing in Codium fragile. Codium fragile is green alga. So,
Yes, sir.
in case of red algae, we see the trends of parasitism and epiphytism also. Some red alga, they have the tendency to deposit calcium carbonate into their cell wall, and such red alga, they are known as coralline algae. That is also unique feature of the red alga, that they have the tendency to deposit calcium carbonate into their cell wall, and thus their thallus become very hard to touch, and they are known as calcareous algae.
Yes, sir.
Reproduction takes place by vegetative and asexual means. Vegetative reproduction. In case of vegetative reproduction, some red algae, they form gemmy. Gemmy-like structure, as we have seen in Bryophytes markensia. Gemmy are reported from the freshwater species Hildenbrandia rivularis. The propagules resembling the spermatangial branch occurs on male and female and tetrasporangial branches of the Polysiphonia. They also form the propagule-like structure, means the structure which we cannot define whether it is formed from the asexual or sexual reproduction. We consider it as the gemmy, or the structure which is a part of vegetative reproduction. Hook-like propagules, which are known as tendrils, and stellate propagules, they are known to found in genus Hypnea, Muciformis, and Hypnea valenti. So these structures, that is gemmy and propagules, they are known to occur in some genera like Hildenbrandia, Polysiphonia, and Hypnea. Sexual reproduction takes place by the formation of the endospores. There are different types of the endospores, which are produced in varying number by special sporangia. These endospores means they are found inside the thallus. They are known as endospores. When the spores are found outside of the thallus, they are known as exospores. So in asexual reproduction, the red algae form endospores, and endospores are of many types, like the monosporangia, bisporangia, tetrasporangia, polysporangia. Monosporangia form monospore, bisporangia form bispores, tetrasporangia form tetraspores, and polysporangia, they form polyspores. It may be formed in gametophytic plant body, or it may develop in sporophytic plant body, or tetrasporangia are found in tetrasporophyte. Right?
Yes, sir.
These are haploid spores, means monospores, bispore, tetraspores, polyspores. All these are haploid spores, and they are produced on diploid thalli after the meiosis and in haploid thalli after the mitosis. Right? अगर डिप्लॉइड थैलाई पे बनेंगे तो हमेशा मियोसिस के बाद बनेंगे. अगर हैप्लॉइड थैलाई पर इनका निर्माण हो रहा है, मींस गैमेटोफिटिक प्लांट पे निर्माण हो रहा है, So these spores are formed after the mitosis because all these spores are haploid in nature, right?
Yes,
The
sir.
sexual reproduction. Sexual reproduction in red algae is oogamous type, always oogamous type, where the male gamete spermacium is non-flagellated and female gamete is carpogonium,
It's
which is a flask-shaped cell with a neck-like protuberance known as trichogyne. Trichogyne was the neck part of the carpogonium.
Yes, sir.
The carpogonia are produced near the apices of the branches, and the carpogonial branch is a two-celled structure consisting of carpogonium cell having trichogyne and a hypogonous cell. So the carpogonial branch is two-celled structure. One is hypogonous cell, and from hypogonous cell there is formation of the carpogonium cell, right? So this is the carpogonium, and this is the hypogonous cell from which the carpogonial branch has developed. This hypogonous cell is also known as supporting cell, or it may develop a separate supporting cell. Hypogonous cell, carpogonial cell, and supporting cell.
Yes, sir.
The hypogonous cell is lateral in position. ये lateral position में है, but it is here in this diagram. It appears that it is just below the carpogonium. Because हाँ, क्योंकि इसका सेल जो है elongated structure होता है, so we can see it in lateral in position. The hypogonous cell is lateral in position and remain attached to the carpogonium and supporting cell through the pit connection. Right? Pit connection set as that time. The carpogonium is conical in shape, bears elongate and swollen trichogyne. This is the trichogyne structure. The egg nucleus is restricted to the basal part. So here you can see this is the egg cell, rounded structure. The male spermatangial conceptacles are present in subcortical region. Means the male branches, they are present in subcortical region of the thallus, and they are irregularly ovoid in shape. Spermatangial conceptacles are irregular ovoid in shape, and they are surrounded by thick mucilaginous covering, giving a swollen appearance to the epidermis. The water motion plays an important role in fertilization of the red algae. So the fertilization takes place with the flow of water or motion of the water or the water current.
Yes, sir.
The wall of both the spermacium and trichogyne are dissolved at a point of contact when, along with the current of water motion, when a spermacium comes in contact with the trichogyne, it always attaches to its neck region or tip region, where a spermacium gets attached. And when it is attached at the point of contact, the cell wall dissolves and spermacium nucleus passes down to the trichogyne and fuses with the nucleus of the egg cell, which is present into the basal part of the carpogonium, and thus zygote is formed. The zygote does not divide directly into spore as in Bangiophysidae. Bangiophysidae is a primitive group of the red alga. In Bangiophysidae group of red algae, the zygote directly divides and it forms the gonymoblast filament. But in other group, means the higher red algae like Floridiophysidae, in Floridiophysidae the zygote take rest and it produces the filaments known as gonymoblast filament after some time or after taking rest, and the terminal cell of this gonymoblast filament form carposporangia. So this is the carpogonium. From this carpogonium, after fertilization, there is formation of the gonymoblast filaments. These are the gonymoblast filaments. Terminal cell of the gonymoblast filament is carposporangium, and inside the carposporangium there is formation of carpospores. Right. So in the higher red algae, the gonymoblast filaments do not arise directly from fertilized carpogonium, but from special auxiliary cells, which may be situated in the carpogonial branch itself. Again, I told you earlier that after fertilization, the carpogonium gives rise to the gonivoblast filament. But still in further some higher group of the red algae, the entire carpogonial branch means the branch consisting of supporting cell, the branch consisting of auxiliary cell, and the branch consisting of carpogonium at its tip. All these cells, they get fused. Here you can see the structure. This is the carpogonium, and just below the carpogonium is hypogynous cell. Hypogynous cell, below the hypogynous cell is supporting cell. So after the
Supporting cells.
fertilization, the supporting cell, hypogynous cell, and carpogonium, all these cells, they fuse together to form a structure, a complex structure. This gives rise to a particular structure known as auxiliary cell. And this auxiliary cell forms the gonimoblast filament, right? So the auxiliary cell is formed after the fertilization and fusion of the cells present into the carpogonial branches, right? So all the cells of carpogonial branch which bear the carpogonia, not all but most of them, they get fused together to form auxiliary cell, and from this auxiliary cell there is formation of the gonimoblast filaments, right?
Yes.
These gonimoblast filaments, they get surrounded by the gametophytic cells, other gametophytic cells, which are meant for the protection of this carposporophytic generation, and this structure is known as pericarp, right? So the pericarp is the protective covering of the carposporophyte or the gonimoblast filament, right? So pericarp is the protective covering of the carposporophytic structure, and the whole structure means the pericarp plus gonimoblast filament, carposporangia, carpospore, all together they are known as the cystocarp, right? सिस्टोकार्प कैसे बनता है? सिस्टोकार्प बनता है जब जो डेवलपिंग गोनिमोब्लास्ट फिलामेंट्स डेवलप हो रही, इनके चारों, so this is the gonimoblast filaments with carpospores and carposporangia. This whole structure is protected by another gametophytic structure known as pericarp. Pericarp is the gametophytic structure, not sporophytic structure. So this gametophyte wants to protect the carposporophyte by making a covering known as pericarp. So pericarp is the covering around the carposporophyte, and the whole structure pericarp plus carposporophyte together known as cystocarp, right?
Yes, sir.
In case of Floridae, the post-fertilization changes resulting in the production of the carposporophyte, which is parasitic on the gametophyte. As I told you earlier, carposporophyte is always dependent on the gametophyte for its nutrition. There is a distinct stage in post-fertilization activities, such as formation of the gonimoblast filaments, either from the fertilized carpogonium or from the auxiliary cell. In primitive forms, the gonimoblast filament develop from carpogonium. In advanced forms, the carpogonium, the supporting cell, and other cells, they fuse together to form auxiliary cells, and this auxiliary cell give rise to the gonimoblast filaments. And this is very significant. This is very significant in case of red algae because it forms the basis for the classification of red algae into the different orders. कि जो कार्पोगोनियम है किस सेल के साथ फ्यूज होकर के गोनिमोब्लास्ट फिलामेंट्स बना रही है, ये इतना इंपॉर्टेंट और इतना प्रोमिनेंट फीचर है रेड एल्गी का कि ऑन दिस बेसिस द ग्रुप Floridiophysidae has been divided into various orders where the post fertilization changes play important feature into the life cycle of these red algae, and it is peculiar for a particular group. Here you can see the life cycle of a typical red algae, that is the haplobiontic. Haplobiontic example, as I told you, the batrachospermum, right?
Yes,
so this is the batrachospermum gametophytic plant, haploid, right? Here in this haploid plant there is formation of the spermatia. In case of Anthridia, you should read it as spermaceum.
Spermatia.
And spermatia, the male plant, and the female plant produces carpogonium. So the
Carpogonium.
spermaceum and carpogonium, here there is fusion of the spermaceum with tip of the carpogonium, right? And
Yes.
after the fertilization, the zygote is formed, and this zygote is formed inside the carpogonium. From carpogonium, there is formation of the goniumoblast filaments. So these are the filaments which are coming out from this gonioblast, from this carpogonium. And these gonioblast filaments, they produce carposporangium. See, this is the carpogonium.
Carpospore.
From this carpogonium, after fertilization, there is formation of numerous filaments known as gonioblast filaments. Tip of the gonioblast filament convert into sporangia called carposporangium. This carposporangium produce carpospores, and this carpospore produce, after germination, produce prothallus-like structure. In some species, this prothallus-like structure produces a special stage which is heterotrichous in nature, known as Catrantia stage. So
Yes,
this is
sir.
the Catrantia stage, intermediate stage. This Catrantia stage, it produces monospores, and this monospore germinate into the new gametophyte,
Mhm.
right?
Yes, sir.
So this carpospore, they themselves, they are haploid. They produce haploid Catrantia stage, which is a filamentous structure. And this
Yes.
Catrantia stage later develops into the adult gametophyte. So one stage is the gametophytic stage, which is haploid. Carposporophytic phase, which is also haploid in nature. And this carposporophyte is dependent on the gametophyte.
Yes, sir.
So this is the haplontic stage where the haploid phase is repeated 2 times. Diploid phase is represented only by the zygote. This is the life cycle of Batrachospermum haplobiontic. In case of diplobiontic life cycle, which is found in advanced algae, here you see the gametophyte. Gametophyte
Yes,
produces the carpogonium and spermatangia. Carpogonium produces, here carpogonium produces carpogonium and egg cell, and spermatangium produces spermaceum. Fusion. After fusion, there is fertilization. This is the carpogonium. It produces carposporangium and carpospore, but without meiosis. जाइगोट में meiosis नहीं हो रही है, इसलिए यहाँ पर जो कार्पोस्पोरोफाइट है वो भी डिप्लॉयड है. क्या है? वो डिप्लॉयड है. So the carpospores which are being produced by the carposporangium, they are diploid in nature. These carpospores germinate to form another generation known as tetrasporophyte. Tetrasporophyte are morphologically similar to the gametophyte. These tetraspores, they produce tetrasporangium inside their thallus. After the meiosis, the tetrasporangium produces haploid tetraspore. Out of 4 tetraspores, 2 convert into the male gametophyte and 2 convert into the
female
so
gametophyte.
the diploid phase is represented 2 times, one in carposporophyte form and another in tetrasporophyte form. So this is the life cycle of diplobiontic. Economically, red algae are very important. 2 important chemicals, they are basically polysaccharide in nature, are derived from the red algae. One is agar and another is carrageenans. Agar is phycocolloid of the red algae. The important property of the agar is it is insoluble in cold water, but it is readily soluble in hot water. If you want to dissolve agar into cold water, it will not dissolve. But if you boil the water and or raise the temperature of the water up to 60 to 80 degrees centigrade, then in such hot water, agar will readily dissolve. And after cooling, it forms gel-like structure at 32 to 39 degrees centigrade. Right. So agar consists of two polysaccharides, agarose and agaropectin. These are the two principal component polysaccharide of the agar, and this agar is obtained from a very common algae, geladium. Geladium is the main source of the agar. After the geladium, pterocladia, acanthopeltis, Anfeltia, and gracilaria. These are the other species from which we get the agar. But the main red alga from which we get the agar is the geladium. So all the red alga which are used for the production of the agar, they are known as agarophytes, right? Sometimes a short note may be asked on what are the agarophytes. So agarophytes are the red algae from which we obtain a complex polysaccharide known as agar. Agar is made up of agarose and agaropectin. It is a phycocolloid which is soluble in hot water, insoluble in cold water. And what is the use of agar? The greatest use of the agar is in association with the food preparation and technology, particularly the culture of the bacteria, the fungi, culture of the fungi. We use agar as a solidifying medium, and it is also used in pharmaceutical industry. It is used for gelling and thickening purpose, particularly for canning of the fish and meat when we transport fish and meat from one country to another. It helps to reduce the undesirable effect of the can. Can में भर के हम लोग ले जाते हैं ना, जिसमें कि पैकेज्ड फूड होता है. तो अगर आप टीन के कैन में किसी चीज को भर के ले जाएंगे तो उसमें टीन के कैन का जो है क्या ना रस्टिंग होती है और वो चीज खराब हो जाती है. तो उसको रोकने के लिए हम लोग अगार में पैक करके इसको भेजते हैं, particularly the eatables, fish and meat. It is also used for making cheese, mayonnaise, pudding, cream, jellies. Agar is also used pharmaceutically. It is used as a laxative, but more frequently it serves as an inert carrier for the drug products where the slow release of drug is required. कई बार दवाएं, कुछ दवाएं ऐसी होती है जिनका स्लो रिलीज होना चाहिए बॉडी में. अदरवाइज आपने दवा खाई और पेट में गया तुरंत डिसॉल्व हो गया तो वो नुकसान कर जाएगा. कई बार होता है कि हमें स्लो रिलीज चाहिए ड्रग का, तो हम उस ड्रग को वी कोट इट विद द अगार एंड गिव टू द पेशेंट्स. It takes time to dissolve and thus helps in slow release of the drug. It is also used in making cosmetics for the skin preparation, ointment and lotion. Agar is also used for bacteriological and mycological culture media. So we can see that there are so many uses of the agar, but agar is a costly stuff.
Yes, sir.
The substitute of the agar is carrageenans. Notice the spelling of the carrageenans. The term carrageenan is derived from a village of Ireland known as Carrageen. Carrageen is a place or a village in Ireland from where the carrageenan harvesting industry is established. The carrageenan is similar to the agar, but it has higher ash content. इसमें राख की मात्रा ज़्यादा होती है. It is obtained from the Chondrus crispus, Gaigartina stellata, Eucheuma, Kappa ficus. These are the red algae from which the carrageenans are used. They are extensively used for the same purpose. We use agar. It is of inferior quality than the agar, but it is cheaper than the agar also. So in all these industries that we discussed for the agar, carrageenan has substituted the agar because of its lower cost and high availability. It is used in medicine industry, canning industry, making ice cream, puddings, and as a thickener, stabilizer of emulsion of the paints, cosmetics, and all these things. Where the agar is used, we can use carrageenans, which is a cheaper substitute of the agar.
Agar.
It is the carrageenans. It is also a sulfated polysaccharide, which is obtained from the cell wall of the red algae. So this is all about the general characters of the red algae. Focus on the reproductive structures. and
sir.
the reproduction in red algae, along with other important characteristic features like absence of the flagella, presence of the pit connection between the
Yes, sir.
adjacent cells, the habitat. These are the important things where you should focus and notice.
Yes,
And
sir.
life cycle also.
Yes, sir.
This is all about the red algae.
Red
Tomorrow
algae.
we will discuss the cyanobacteria or blue-green algae in the class.
Yes, sir. So tomorrow also you'll take class the online only?
No, tomorrow
Okay.
I'll be in the college.
Okay, sir. So we'll come.
Mhm.
Okay, sir.
Okay, this is all for today.
Okay. Thank you, sir.